Okay, thank you very much. So, as Estelle has already said, my name is Thomas Tscheulin. I'm from the University of the Aegean in not so sunny Greece at the moment. And I will quickly talk you through the general process of screening the literature. So, I am part of the multidisciplinary steering group. As we've already heard, there were 4 experts on different taxonomic groups. So, yesterday we heard from Alain Villon on plants, and also from Pascal Malkemper on invertebrates, on the vertebrates. And today you will hear from me and from Adam Vanbergen on the state of the invertebrates. Apart from the ecological or biological experts on those taxonomic groups I just mentioned, we had also 2 technical experts who looked at the technical side of those publications we looked at. They identified 15 categories of radiation types that I will show you in a minute. Then we went on to look at the literature, newer literature, so after 2000, published after 2000, focusing on the scientific literature, peer-reviewed research only, and also some reviews. The scope of our screening was to look at the effect of EMR on wildlife only. So we're not looking and not talking about human health, and there were only a very limited number on domestic animals we looked at, but we didn't take those into account. The EMR range we looked at in those publications was only artificial anthropogenic radiations, and the light was excluded from the electromagnetic radiation. So this left us with an initial 147 identified publications, and it was narrowed down by these restrictions that we have to final 97 papers, all on those 3 groups: plants, invertebrates, and vertebrates. So I'll only talk about the invertebrates today. Oops, I went one too far. So on the right-hand side in this box, you see the 15 different fields that were identified by the physicists, by the 2 technical experts. I will not go further into detail on those. And then we split up into these groups. So Adam and I were dealing with the invertebrates, and we've already heard about the plants and vertebrates. The technical expert looked at the EMR ranges, and the taxonomic experts then looked at the studies themselves to look how well they were conducted and what the main findings were. First, we decided whether the studies were field-based or lab-based, or maybe both. And then we also looked at the quality of the studies. So we had 4 categories: the bad or insufficient quality studies, which we did not take into account further. We just scored those. And then we had studies that were of minimum quality, which means that they had big flaws, those studies, but there were still some elements that we wanted to take into account. Then mostly the medium or normal quality studies with some shortcomings, as studies usually are anyway. And then there were some few very excellent quality studies where we couldn't fault the studies on the grounds of the setup, which doesn't necessarily mean that these are actually the studies that advance us a lot in our understanding. Very often the excellent studies were studies that had a very specific focus and did not have a broad finding that kind of advanced us, but I will go into detail in a little bit. In the second step, we then try to extract the main findings from those publications we looked at, and also to find the knowledge gaps. So this left us with 39 publications on invertebrates that we looked at. The diagram you see here, the table, is actually the outcome of the technical aspects, so the technical experts that looked at them. They managed to look at only, due to time constraints, only managed to look at 26 out of the 39 publications on invertebrates. And they scored 7 out of those 26 they looked at as very bad quality or technically irrelevant. 2 were of minimum quality as far as the technical aspects go, and 5 were of normal quality, and 12 were deemed excellent by the 2 experts. This is only on the level of the EMR on how the radiation was actually presented and how the exposure was done from a technical point of view. So mine and Adam's work was then to look at the studies from a biological, ecological point of view. So we looked at all 39 papers, publications. We deemed 3 completely irrelevant because they were anecdotal, basically. There was no statistical analysis, or they were not. You could not replicate those studies because the description of the study was so bad that it was hard to understand how they actually did it. Then 10 studies were minimum quality, but some elements were still useful for our screening here. And 17 were kind of a normal quality. This is the majority here, and some excellent quality studies as well. Okay, I want to make some general assessment first as an overview of what we found. Most of the studies, most of those 39 studies dealt with model insects, so they were mainly on honeybees and Drosophila, and some cockroaches as well, but they were all kind of model insects mostly. And as a result of this, all studies had a very limited scope, so mainly dealing with a single species, and mainly also dealing with those species within the lab. So there were only 3 field studies out of these 39, and not all of those actually dealt with anthropogenic EMR. So many of those 39 studies actually dealt with natural fields, magnetic fields. We found that there was almost complete lack of ecological community studies. So it's just one community study that is worth mentioning here out of those 39. The majority of the laboratory studies focused on some physiological or developmental aspects of the specific model invertebrate that was used. Mostly insects, just 1 or 2 studies on crustaceans, but mostly the invertebrate are actually covered by the insects only, so they were mostly only insect studies. And most of those studies are purely short-term acute exposure studies. So basically the insects were, or the invertebrates were subjected to a very short exposure to EMR, and then the effects were measured afterwards. It was just one study which examined long-term effects on invertebrates, and this study was also a field-based study. I myself was actually a co-author of this study. I will tell you a little bit more in a little bit. So some more specific problems with the 39 publications or criticisms. There was very mixed quality, as I already mentioned. Some had very low replication levels, some were not replicated at all. This makes them anecdotal and not of scientific interest. And quite a few had a complete lack of statistical analysis as well. The field studies, 3 of them, suffered from some confounding effects, as is usual for field studies, unfortunately. The lab studies were often not transferable to real-life situations, given the very short-term exposure to the EMR, which is not usually the case in real life. So usually, if there's radiation, this is something that is ongoing and will have an effect over several generations, so it's not a short thing. So that lab studies usually very often oversimplify the facts. The limited scope was a problem. As I said, there was only selected species in most cases, and only a very short-term exposure. And sometimes the exposure to EMR was very unrealistic in terms of time and space. So very short-term, and also very strong EMR in some cases. So, for example, several studies placed mobile phone handsets in or near the insects or invertebrates, and then called those devices, or had decked phones very close, like in beehives. And this is a type of exposure which is highly unrealistic in real-life situations. Another problem was that the studies were not very comparable. So out of those 39 studies, there were no 2 studies that actually had a similar setup. So methodology was very different, which means that the outcome of those studies cannot be easily compared, which is a big problem. Right. I want to quickly talk you through, Estelle has already done it, but I will quickly talk you through this model we used for actually placing the findings in terms of the merits of those studies. So you have two axes on this diagram. On the x-axis, you see the quantity and quality of and the evidence presented. And on the y-axis, you see the level of agreement between the studies. And ideally, we would like to be in the top right corner. So we want evidence to be well established, which means that we have good quality evidence and we have good quantity evidence, which means several studies have found the same thing. And if this evidence agrees with each other, so if we have the studies actually agree with each other, then we are in the top right corner and we have well established facts, findings, which is the ideal case. Unfortunately, we did not have this in any of the findings, as you will see in a little bit. The worst case scenario is to have inconclusive findings, which means that either the quality of the study having this finding is very bad, or the quantity is very low, or you have several findings that disagree with each other. Okay, I will quickly talk you through the main findings that we saw in those 39 publications. Number one is that some invertebrates have a magnetic sense. We've heard that also for the vertebrates. This is a finding that is established, but it's incomplete because there was only a few species that those studies looked at. And there's quite a high level of agreement between those studies, but there are only a few species. So there's a problem of quantity in this case. Then this magnetic sense seems to have been affected in some cases by EMR, and several studies have shown this. Quite often they disagree with each other, these studies, but I'll give you a few examples. So there were a study on ants, and this study showed that some changes in the animal's locomotion took place. So there was, for example, a lower response to the alarm pheromone in ants, or even a lower response to the trail pheromone, which means that the ants actually got lost when they were exposed to EMR. Then we have a study on honeybees, on Apis mellifera. They showed that there was a reduced motor activity of the worker bees, so they didn't move in the same way that they didn't move before. Also, locomotor activity was affected by Drosophila subobscura after exposure, and as well in locusts, study found that the exposure to some electromagnetic radiation treatments affected the walking, so locomotion was affected again. In this case, we have quite a bit of disagreement between the studies as well, and we don't have enough good quality studies, so we deemed this effect and this finding inconclusive. So if you go to the next finding, one finding is that there's some evidence that anthropogenic EMR can affect reproduction, and we had several studies on this. Only two actually found an effect. So this effect seems to be, this finding seems to be highly in disagreement between the studies. There were two studies which found a negative effect, and they both are about Drosophila melanogaster, so Drosophila flies. In both of those studies, the reproduction was affected. In one case, the egg to the imago development period and the imago longevity was affected. It was reduced or increased depending on the level of radiation. And another study also on the Drosophilas found that mobile phones decreased the reproductive capacity of Drosophilas by almost half. But as I said, there were several other studies who did not find any effect. So we deemed this that there's some disagreement, and that's why we deem this specific finding as unresolved. We need more. We definitely need more support for this specific finding. And then we have, so far these studies have only dealt with actually single species studies. And when we look at the population and community studies, there's actually only one. And as I said, I I myself was a co-author of that, so I can give you a little bit more background on this. There's only one study, which means that by default it cannot be established, the findings that we have in there. There's only one study. There cannot be any disagreement if there's only one study. So by default, we have inconclusive information in this case for this specific finding. There's no conclusive evidence. What we found in this study, it was carried out in Greece on 2 islands. We used 15 mobile phone antennas, and we measured abundances and species richness of several pollinators at different distances from those antennas. We measured the EMR intensities, and we tried to relate this to the sampling we had from different species of pollinators. So we looked at wild bees, hoverflies, bee flies, beetles, butterflies, and wasps, and looked at abundances and species richness of those, and then looked at if those levels are in any way related to the intensity of the EMR. And we actually found effects in all groups apart from the butterflies. All groups were affected by EMR. The beetles, wasps, hoverflies, abundance decreased with EMR, so it was a drop in EMR if there was a higher intensity of EMR. There was a drop in abundance if there was a higher intensity of EMR. And it was quite interesting to see that the abundance of underground nesting wild bees and bee flies, they're both underground nesting, they actually increased with EMR. So it seems to be contradictory, but our take was that maybe underground nesting insects are protected. At least the developmental stages of the larvae are protected from the EMR, and maybe they actually taken advantage of lower levels of other competitors, and that might make them kind of more numerous, more abundant in case they're missing. But there's just this one study of community level insects, and we cannot call this conclusive because it's geographically quite limited, and also just one study. So quickly, if we place this on the grid, we see that unfortunately we have no finding which is well established. Some is unresolved, as I already said. Some is completely inconclusive because there's a lack of good quality studies and quantity as well. And some we have some level of agreement between the studies, but the quality and the quantity is not enough. So I quickly want to talk you, and this is my last slide, quickly want to talk you through the knowledge gaps. We definitely need more field ecological studies measuring the effects of EMR. We need community responses to EMR, long-term and ideally pre and post-exposure, and over a wide geographical extent, and not only in Greece as our study is, and also in different natural and anthropogenic systems. We definitely need interdisciplinary collaborations. We had in our group, we had actually a physicist dealing with the EMR. I think that this is essential because I'm a biologist. I have no idea about physics. We need testable hypotheses, and these studies need to be based on this, and which should not, as in some of those 39 studies, just look for any differences without actually having a hypothesis from the start. We need field realistic exposure, so no extreme exposure, or no overexposure, and no close distance exposure, which is not realistic in the field. We need more studies assessing the chronic effects and not these short-term effects. We need more comparable data, so we need some common methodology, and we also need to look at maybe some indirect and interplay of EMR exposure on other aspects like foraging ecology or other stresses affecting nutritional ecology. So I think that's all for me.